வாகை சூடவா.
Dear students, now I like to give a brief introduction about pollination. You know pollination is nothing but transfer of pollen grain from anther lobe to the stigma of the carpel. Transfer of pollen grain from the anther lobe. Anther, part of the stamen, transfer of pollen grain from this anther lobe to the stigma of the carpel. This is carpel with ovary, style and stigma. Transfer of pollen grain or microspore from this anther lobe to the stigma of the carpel is said to be pollination, and this pollination followed by fertilization, you know. Two types of pollination reported in higher plants, especially the protected seeded plant, especially the angiosperms. One is self-pollination or autogamy. Another is the cross-pollination or xenogamy or allogamy. Transfer of pollen grain from anther lobe to the stigma of the carpel of the same flower is said to be self-pollination or autogamy. Transfer of pollen grain from anther lobe to the stigma of the carpel of the another flower present in the same species, a different plant, another plant of the same species, is said to be cross-pollination. Transfer from anther lobe to the stigma of the same flower is said to be self-pollination. Transfer of pollen grain from anther lobe to stigma of another flower present in another plant of the same species is said to be cross-pollination. These things you know. Then cross-pollination plays a vital role in origin of new species, and the agents responsible for cross-pollination, agents or agencies, one is the abiotic, another is the biotic. Abiotic components helps in cross-pollination or the wind and water. The pollination through wind is said to be anemophily or wind pollination, and pollination with the help of water is said to be hydrophily. This anemophily pollination common in gymnosperms. Naked seeded plants are defined as gymnosperms, or seeds without covering gymnosperms where anemophily is predominant. Hydrophily, pollination with the help of water seen in the flowering plants, especially the hydrophytes, submerged hydrophytes. The biotic agencies, the biotic components helps in cross pollinations, or you know the different types of cross pollination with the help of animals: canthrophily, felanophily, malacophily, psychophily, ornithophily, chiraptrophily, and finally myrmecophily. Pollination with the help of ants is said to be myrmecophily. Pollination with the help of bat is said to be chiraptrophily. Pollination with the help of birds, the aves, is said to be ornithophily. Pollination with the help of snails is said to be malacophily. Pollination with the help of butterflies is said to be psychophily. Pollination with the help of bees is said to be melatophily, and pollination with the help of moth is said to be felanophily, and with the help of beetle is said to be canthrophily. All these biotic components, living organisms, helps in cross pollination. Then contrivances of cross pollination, or the things helps in cross pollination: number 1, protandry; number 2, protogyny; number 3, distyly; and number 4, tristyly. You know protandry. If androecium matures before gynoecium is said to be protandry, favors cross pollination. If gynoecium matures before androecium is said to be protogyny, it also helps in cross pollination in higher plants. Like that, the style type, you know, style, this is style, ovary, style and stigma. If the plant possesses two different type of styles, we are calling it as distyly, seen in primula. and 3 different types of styles present in plant. We are calling it as tri-styly seen in Latharum. Then this double fertilization and triple fusion favors cross-pollination as well as fertilization in higher plants. What is fertilization? The fusion of male and the female gametes, and it results in the formation of a diploid cell called zygote is said to be fertilization. Fusion of male and the female gametes, and it results in the formation of diploid cell called zygote. The process is said to be fertilization. In flowering plants, double fertilization and triple fusion takes place. At the time of microsporogenesis, development of microspore or the pollen grain from the microspore mother cell is said to be microsporogenesis. Microspore is always haploid in nature. This microspore is the end product of microsporogenesis. From this microspore, 2 male gametes are formed. Not straight away from the microspore. From the microspore, generative and the vegetative cell is formed. From the generative cell, 2 male gametes are formed. The male gametes are also haploid in nature. The development of megaspore from the megaspore mother cell is said to be megasporogenesis. This megaspore is haploid in nature. This megaspore undergo nuclear division to form the female gametophyte or the female gametophyte is also known as the embryo sac. This embryo sac is always in an 8-celled stage, antipodals, the polar nuclei in the middle, and the egg apparatus. This cell is very important. This is said to be the egg. These 2, the polar nuclei N plus N, fuse to form the secondary nucleus. The secondary nucleus is diploid in nature. 2N, 2 sets of chromosome. The egg with N, haploid, only 1 set of chromosome. What is double fertilization and triple fusion? From the generative cell of the microspore, 2 male gametes are formed. 1 male gamete fuses with the egg to form diploid zygote. N plus N is equal to 2N. N plus N, the 2N, diploid zygote. This is one fertilization. Another male gamete fuses with the secondary nucleus. The secondary nucleus is diploid in nature to form triploid endosperm, or we are calling it as primary endosperm nucleus, 3N. Here 1 male gamete fuses with the egg to form diploid zygote, 1 fusion. Another male gamete fuses with the secondary nucleus which is diploid in nature to form triploid endosperm. So 2 male gametes are involved, 2 fusions, 2 fertilization takes place. So it is said to be double fertilization. Why it is known as triple fusion? See here, first 2 polar nuclei undergo fusion. N plus N is equal to 2N secondary nucleus. 1 male gamete fuses with the egg, another fusion. Another male gamete fuses with the secondary nucleus, third fusion. So double fertilization is also known as triple fusion, results in the formation of diploid cell called the zygote. This zygote undergoes successive mitotic cell division and develops into the embryo. This primary endosperm nucleus undergoes cell division. It is also mitotic in nature to form the endosperm. Endosperm is a triploid one. Generally, the plant body is diploid. The gamete is haploid. The only triploid part seen in higher plants means this endosperm. Tender coconut is a very good example for endosperm. What is the role of endosperm? The main function of endosperm is to supply nutrients to the developing embryo. The developing embryo present inside the seed with radicle plumule and cotyledon, they derive nutrients only from this triploid endosperm. The next one is this types of endosperm. So endosperm, you know, it is the end product of double fertilization and triple fusion. What is the role of endosperm? The main function of endosperm is to supply nutrients to the developing embryo. Actually, three types of endosperms reported in higher plants. One is nuclear endosperm, second is cellular endosperm, and third is halobial endosperm. Nuclear endosperm, I hope you know two terms in biology. One is karyogynesis, the division of nucleus. The second one is cytokinesis, division of cytoplasm. At the time of mitotic as well as meiotic cell division, the nucleus undergo division first. So nuclear division, karyogynesis, the division of cytoplasm is said to be cytokinesis. You also know the term karyogynesis like that, karyogamy and plasmogamy. Karyogamy means fusion of nuclei. Plasmogamy means fusion of protoplast. Now scientists through protoplasmic fusion produce interspecific somatic hybrids. This is fusion, this is division. So three types of endosperms. One is nuclear endosperm, another is cellular endosperm, third is halobial endosperm. Nuclear endosperm means, take this is the primary endosperm nucleus, pen. In this kind of endosperm, the nucleus alone undergo division. Karyogynesis only occurs here. So it results in the formation of endosperm in a multinucleate condition. In the 11th standard, you read this multinucleate condition in fungi. Three types of mycelium reported in fungi. One is called the monokaryotic mycelium, second is the dikaryotic mycelium, and third is the cenocytic mycelium. The cells present in the hypha with only one nucleus, monokaryotic. The cells present in the mycelium or hypha with two nuclei, dikaryotic. Multinucleate aseptate mycelium is said to be cenocytic mycelium seen in fungi. Like this cenocytic condition, if the endosperm shows the cenocytic condition or the endosperm with many nuclei, thus nuclear endosperm. Cellular endosperm means karyogynesis followed by cytokinesis takes place. In this kind of endosperm, the primary endosperm nucleus undergo karyogynesis followed by the division of cytoplasm. This kind of endosperm is said to be cellular endosperm. Halobial means first the pen undergo nuclear division, two nuclei are formed. In one side of the nucleus, cytokinesis takes place. In the opposite side, only karyogynesis. This lower part continuously undergo karyogynesis and cytokinesis. So in the lower side, the endosperm is cellular in nature. In the upper side, it is cenocytic or nuclear in nature. This kind of endosperm is said to be halobial endosperm, and then ruminant endosperm is also present. Certain endosperm, the outer surface is irregular in nature. This kind of endosperm is said to be ruminate. So four different types of endosperm. These three are predominant in flowering plants. The next one is seed. After fertilization, the egg is converted into zygote. The secondary nucleus is converted into the endosperm or the primary endosperm nucleus. The diploid zygote undergoes successive mitotic cell division to form a structure called the embryo. So embryo is the end product of mitotic cell division, and the embryo with 3 parts, namely number 1, the radicle, embryonic root. Number 2, the plumule, embryonic shoot. In between the radicle and plumule, the cotyledon is present. Embryonic root is said to be radicle, embryonic shoot is said to be plumule. In between the radicle and plumule, cotyledon is present. The region between radicle and cotyledon is known as hypocotyle, and the region between cotyledon and the plumule is known as the epicotyle. This is the structure of dicot embryo. So dicot embryo, radicle, it develops into the embryonic root. This is plumule, on germination develops into embryonic shoot. On the basis of development of hypocotyle and epicotyle, 2 different kinds of germination seen in plants. One is epigeal germination, another is hypogeal germination, the third one, very rare one, the viviparous germination. What is hypogeal germination? Hypogeal germination means if epicotyle grows faster, then cotyledon remain buried inside the soil. This kind of germination is said to be hypogeal germination. If the hypocotyle grows faster, then cotyledon comes out of the soil, it is said to be epigeal germination. See here, if hypocotyle grows faster, then cotyledon comes out, this is called epigeal germination. If epicotyle grows faster, then cotyledon remain buried inside the soil, this germination is said to be hypogeal germination. The plants grow in marshy land, especially mangroves. The germination takes place in the plant itself. If germination takes place in the plant itself, it is known as viviparous germination, unique feature of the mangrove trees. The plants live in marshy land. Then the seed with the embryo, and the embryo surrounded by 2 outer coverings. The inner covering is said to be tegumen, and the outer covering of the seed coat is said to be testa. The outer coverings of the seed coat or the seed, outer seed coat is known as the testa, and the inner seed coat is known as tegumen. This is regarding the dicot embryo. But monocot embryo have some special structures called the coleorhiza and the coleoptile. Now we come to the structure of monocot embryo. See here, this is monocot seed. This area, the embryo is present. The embryonic root of monocot is said to be radicle. The embryonic shoot is said to be plumule. The cotyledon of monocot is known as the scutellum. Monocotyledonous plants have only one cotyledon. The cotyledon is also known as the scutellum. And this area, the endosperm. The radical portion of the embryo surrounded by coleorhiza, and the plumule portion of the embryo surrounded by coleoptile. These structures we should remember regarding monocot embryo. Then post-fertilization changes. What are the changes takes place in the ovary or in the carpel or in the gynoecium after fertilization? Point number 1, what will happen to sepals, petals, stamens in carpel? Sepal, petal, stamen, style, stigma usually wither off. Sepal wither off or falls, petals, stamen, style and stigma no use. The ovary is converted into fruit. After fertilization, the ovary is converted into the fruit wall or the pericarp with 3 parts, namely epicarp, mesocarp and the endocarp, and the ovules. the ovules are converted into seeds. After fertilization, ovule is converted into seed and the ovary is converted into fruit wall or the pericarp. That's why the fruit is said to be fertilized, ripened, matured ovary. Then egg is converted into zygote. Egg is converted into zygote. Then secondary nucleus is converted into the endosperm or primary endosperm nucleus, pen. Then antipodals disintegrates, synergis disintegrates, outer integument is converted into testa, inner integument is converted into tegument. The funicle, funicle, stalk of the ovule is said to be funicle, it is converted into stalk of the seed. Like that, micropyle of the ovule is converted into the micropyle of the seed. Very important change, ovule is converted into seed, ovary is converted into the fruit wall or pericarp, egg is converted into zygote, secondary nucleus is converted into pen, primary endosperm nucleus. Then summary of the entire reproduction, entire sexual reproduction. See the flower, the fertilized, ripened, matured ovary. Fertilized, ripened, matured ovary is said to be fruit. Flower is a modified condensed shoot meant for reproduction. So flower is a modified condensed shoot meant for reproduction, and fertilized, ripened, matured ovary is said to be fruit. The male reproductive organ present in the flower is said to be androecium, formed by the union of stamens, and the female reproductive structure is said to be gynoecium, formed by the union of carpels. From the stamen or the microsporophyll, after meiotic cell division, microspores or pollen grains are formed, and inside the gynoecium, meiotic cell division takes place from the megaspore mother cell, megaspores are formed, haploid megaspores are formed. It is also the end product of meiotic cell division. From the microspore, generative cell and the vegetative cell is formed. From the generative cell, two male gametes are formed. And megaspore possesses the egg, the haploid egg. Pollination followed by fertilization, fusion of male and the female gametes results in the formation of a diploid cell called the zygote. Zygote develops into the embryo, embryo present inside the seed, seeds on germination develops into the higher plant. So it is a summary of sexual reproduction. The next one is apomixes and polyembryony. Amphimixes. What is amphimixes? Then we come to apomixes. Amphimixes means the development of new organism after gametic fusion. After gametic fusion, new organism is formed, then it is known as amphimixes. A development of new organism without the fusion of gametes or without fertilization is said to be apomixes. So amphimixes, development of a new organism or development of embryo with fertilization or fusion of male and the female gametes. Here fusion of male and the female gametes is absent, fertilization is absent, but a new organism is developed. Then this apomixes without fusion, two types of apomixes reported in higher plants. One is recurrent, another is non-recurrent. The recurrent apomixes is of two types, namely the vegetative reproduction and agamospermy. Vegetative reproduction, the root modification, the stem modifications, the leaf modification or reproduction through the vegetative parts like root, stem and leaf. We are calling it as vegetative reproduction. The vegetative structures develops into a new organism. Vegetative reproduction, the recurrent apomixes type. Agamospermy means gametic fusion is absent. This agamospermy also responsible for the development of new organism, and three types of agamospermy seen in higher plants. One is adventive embryony. the integuments is develops into a new plant body, we are calling it as new plant body or new embryo, we are calling it as adventive embryony. Diplospory, the megaspore mother cell straight away develops into a new organism or embryo, we are calling it as diplospory. Apospory means the megaspore mother cell undergo meiotic cell division to form 4 haploid megaspores. All the megaspores disintegrates, some of the cells present in the ovule straight away develops into the embryo sac, we are calling it as apospory. Then another aspect is polyembryony. Poly means many, embryo means embryony means embryos. Development of more than one embryo after fertilization is said to be polyembryony. These are all the important things you should know in the sexual reproduction. Dear students, now I am going to discuss important questions in pollination, fertilization and post-fertilization changes in flowering plants. Question number 1, the advantage of cleistogamy. You know, cleistogamy is a kind of pollination. Option A, higher genetic variability. Option B, more vigorous offspring. Option C, no dependence on pollinators. And option D, vivipary. First, you know the meaning for the term cleistogamy. Cleistogamy is a kind of self-pollination where the flower never opens at all. So, it favors only self-pollination because the flower never opens. So, pollinators are needed or not needed. Pollinators which helps in pollination, it may be either abiotic or biotic. For this cleistogamy kind of pollination, pollinators are not needed. So, no dependence on pollinators or no dependence on pollinator is the answer for the question. Option A, higher genetic variability only seen in cross pollination. More vigorous offsprings are also formed in cross pollination. Vivipary is not a pollination, it is a kind of germination. Germination of seed takes place in the plant itself, unique feature of mangrove trees, Avicennia and Rhizophora. So, answer for the question number 1, option C, no dependence on pollinators. No dependence on pollinators. Question number 2, pollen tablets are available in the market for option A, in vitro fertilization, option B, breeding programs, option C, supplementing food, option D, ex situ conservation. Pollen tablet, you know, pollen grain is the end product of microsporogenesis. Microspore is also known as the pollen grains. In vitro fertilization means fertilization outside the body of the animal, in the artificial medium, in the artificial environment. Breeding program helps to produce high yielding varieties. Ex situ conservation, ex situ, in situ conservation, you know. Ex situ conservation, conserving the plant or animal in the external environment. So, pollen enriched with the carbohydrates, proteins, vitamins and minerals. So, the correct answer for the question number 2 is option C, supplementing food, because pollen, the microspore, enriched with carbohydrates, proteins, vitamins and minerals. So, the correct answer is supplementing food. Question number 3, geitonogamy involves. Geitonogamy is a kind of pollination. It involves option A, fertilization of a flower by pollen from another flower of a same plant. Option B, fertilization of a flower by pollen of the same flower. Option C, fertilization of a flower by pollen from a flower of another plant in a same population. Option D, fertilization of a flower by the pollen from a flower of another plant belongs to distant population. Geitonogamy is a kind of pollination. The correct answer for the question 3 is option A, fertilization of a flower by pollen from another flower of a same plant. It is also a kind of self-pollination. Question number 4. Functional megaspore in angiosperm develops into an option A endosperm, option B embryo sac, option C embryo, option D the ovule. Functional megaspore. Megaspore, you know, the end product of megasporogenesis. In the end of megasporogenesis, from a single megaspore mother cell, 4 megaspores are formed, 3 megaspores disintegrates, only one is functional. The functional megaspore is nothing but the embryo sac. You know, endosperm. Endosperm is a nutritive tissue for the developing embryo, and it is the end product of double fertilization and triple fusion. Embryo developed from the diploid zygote after mitotic cell division. Ovule is nothing but megasporangium. So the correct answer for the question number 4 is option B embryo sac. The functional megaspore or the megaspore undergoes successive mitotic cell division on its nucleus and form the female gametophyte or embryo sac. And the embryo sac already you read in your book with 8 cells, antipodals, the secondary nucleus, the polar nuclei, and the egg apparatus. So this is the female gametophyte or the embryo sac. So the correct answer is option B, the embryo sac. Question number 5. When a diploid female plant is crossed with a tetraploid male, the ploidy of endosperm cells in the resulting seed is. Read the question once again. When a diploid female plant, diploid means 2 sets of chromosome. When a diploid female plant with 2 sets of chromosome is crossed with a tetraploid male, this is female, crossed with a tetraploid male, male is tetraploid in nature. The question is the ploidy of the endosperm cells in the resulting seed. The ploidy of endosperm. Endosperm is a nutritive tissue for the developing embryo, you know. It is always triploid in nature, but here see the cross. A diploid female, from the diploid female after megasporogenesis, megaspore is formed. The megaspore is a haploid in nature. This megaspore undergo nuclear division to form 8 nuclei. In the middle, 2 polar nuclei is present. This is polar nucleus 1, this is polar nucleus 2. Here it is N, this is N, N plus N is equal to 2N. This polar nuclei become diploid secondary nucleus. This diploid marries a tetraploid male, crossed with a tetraploid male. Tetraploidy means 4 sets of chromosome. This male undergo meiotic cell division to form microspores or pollen grains. The microspores are diploid in nature because in the end of meiosis 4N becomes 2N. From the diploid microspore, diploid male gametes are formed. Here the male gamete is also diploid in nature. This diploid male gamete fuses with the diploid secondary nucleus 2N plus 2N, you get 4N. So the answer for the question number 5 is option D, tetraploid. That means the endosperm of this cross with 4 sets of chromosome, the condition is said to be tetraploidy. Question number 6. Which one of the following pairs of plant structures has haploid number of chromosome? Which one of the following pairs of plant structures has haploid number of chromosome? Haploid means N set of chromosome. Option A, egg nucleus and the secondary nucleus. Option B, megaspore mother cell and antipodal cells. Option C, egg cell and the antipodal cell. Option D, nucellus and antipodal cells. See the option once again. Option A, egg nucleus haploid, secondary nucleus diploid. Option B, megaspore mother cell diploid, antipodal cells haploid. Option C, X cell is haploid, antipodal cell is also haploid. Option D, nucellus is diploid, antipodal cell is haploid. So, the correct answer for the question number 6 is option C, the X cell and the antipodal cells with only one set of chromosome. So, these two shall show haploid condition. Then question number 7, the arrangement of nuclei in a normal embryo sac or the female gametophyte in a dicot plant, 2 plus 4 plus 2, option B 3 plus 2 plus 3, option C 2 plus 3 plus 3, option D 3 plus 3 plus 2. See the board. This is the female gametophyte or the embryo sac. In the posterior side 3 nuclei are present or 3 cells are present known as the antipodal cells. See here the egg apparatus with the 3, here in the middle 2. So, 3 plus 2 plus 3, this arrangement is seen in the female gametophyte or the embryo sac. So, correct option for the question, the correct answer for the question 7 is option B, 3 plus 2 plus 3. 3 represent antipodal cells, 2 represent the polar nuclei, 3 represent the egg apparatus. Then question number 8, wind pollinated flowers are, wind pollinated means pollination takes place with the help of wind. This kind of pollination is said to be anemophilous. Anemophilous kind of pollination common in the naked seeded plants, gymnosperms. Option A, small producing nectar and dry pollen. Option B, small brightly colored producing large number of pollen grains. Option C, small producing large number of pollen grains. Option D, large producing abundant nectar and pollen. Generally, nectar plant or the flowers with nectar attract the insects for cross pollination. Generally, large sized flowers with nectar attract the birds. So, large flower with abundant nectar usually attracts the birds. So, correct option for the question, correct answer for the question number 8 is option C, small flowers producing large number of pollen grains usually favors wind pollination or anemophilous kind of pollination. Then question number 9, the function of filiform apparatus is to A, recognize the suitable pollinate stigma, B, stimulate division of the generative cell, option C, produce nectar, and option D, guide the entry of pollen tube. Question, the function of filiform apparatus, you know filiform apparatus present in the gynosium or in the carpel or in the pistil. The main function of this filiform apparatus to guide the entry of pollen tube. So, it helps in the entry of pollen tube. So, the correct answer for the question number 9 is option D. The function of filiform apparatus is to guide the entry of pollen tube. Then question number 10, the coconut water from tender coconut represent. Coconut is an example for drupaceous fruit. It is a single seeded fruit, and coconut with epicarp, mesocarp and stony endocarp. So, coconut is also a very good example for stone fruit. Coconut, very good example for fleshy fruit. Fleshy fruit, succulent fruit. Succulent here, the mesocarp is not succulent in nature, fibrous. The endocarp is very hard in nature. So, the endocarp, due to the presence of very hard endocarp, it is also known as the stone fruit. The coconut water present inside the endocarp of tender coconut represent option A, endocarp, option B, fleshy mesocarp, option C, free nuclear pre-embryo, and option D, free nuclear endosperm. Endocarp, it is not the edible portion, it is very hard in nature. Fleshy mesocarp, in coconut the mesocarp is not fleshy in nature, it is fibrous in nature. Free nuclear pre-embryo, it is not embryo, and finally free nuclear endosperm. The correct option, correct answer for the question number 10 is option D, free nuclear endosperm. You know, endosperm is a nutritive tissue for the developing embryo, and 3 types of endosperm reported in higher plants. Number 1, nuclear endosperm. Number 2, cellular endosperm. Number 3, helobial endosperm. The coconut possesses free nuclear endosperm, and it is the edible portion in coconut, Coccus nucifera. Then question number 11, pollination in water hyacinth. Water hyacinth, you know, eichhornia, and water lily is brought about by the agency of pollination. Is nothing but transfer of pollen grain from anther lobe to stigma of the carpel. Pollination in water hyacinth, the free floating hydrophytes, and water lily attached floating hydrophyte is option A, insects or wind, option B, birds, option C, bats, and after option D is water. Insect, birds, water and bats. The correct answer for the question is option A, insects or wind pollination takes place in water hyacinth and water lily. See the answer, option A. Then question number 12, perisperm differs from endosperm in. Perisperm differs from endosperm in. Option A, being haploid tissue. Option B, having no reserved material. Option C, being a diploid tissue. Option D, its formation by fusion of secondary nucleus with several sperms or male gamete. See the question, perisperm differs from endosperm. What is perisperm? You know, nucellus tissue present in the ovule supplies nutrients to the developing female gametophyte. After fertilization, the remaining part of the nucellus tissue present in the embryo is said to be perisperm. So, perisperm is nothing but the nucellus. What is nucellus? Nucellus is a nutritive tissue for the developing female gametophyte. It is present in the ovule inside the integuments. So, after fertilization, the ovule is converted into seed, the ovary is converted into fruit, wall or the pericarp. The remaining nucellus tissue present in the embryo or present in the seed in the form of perisperm. So, nucellus is diploid in nature, so perisperm is also diploid in nature. So, the correct answer for the question number 12 is option C, being a diploid tissue, the remaining or the reminiscent of the nucellus present in the embryo. Then question number 13, in a type of apomixes known as adventive polyembryony, embryo develop directly from option A, synergids or antipodals in embryo sac, option B, nucellus or the integuments, option C, zygote, option D, accessory embryo sac in the ovule. The question is in a type of apomixes. You know, apomixes, development of a new embryo, development of a new organism, development of a new offspring without the fusion of gametes. So, apomixes means fusion of gametes is totally absent. So, what are the things possible here? Option A, synergids is haploid in nature, antipodals haploid in nature. So, it is haploid, but the embryo is usually diploid in nature. So, first option is not possible. Second is nucellus or integuments. Nucellus, the nutritive tissue for the developing female gametophyte. Integuments, the outer covering of the nucellus or the ovule. So, the correct answer for the question number 13 is option B, the nucellus or integuments which are diploid in nature without fertilization, without the fusion of gametes, they straight away develops into embryo. This kind of embryo development is said to be adventive polyembryony. Then question number 14, in a cereal grain, a single cotyledon of the embryo represented by, you know, cereal comes under monocotyledonous plant. Monocotyledonous plant, the question is in a cereal grain, the single cotyledon of the embryo is represented by, see the embryo of monocot. This is the embryo of monocot. See this region, this is the endosperm, nutritive tissue for the developing embryo. The embryonic root is said to be the radicle, embryonic shoot is said to be the plumule, the radicle is surrounded by coleorhiza, The plumule is surrounded by the coleoptile. This area is said to be cotyledon. The cotyledon of monocot is said to be scutellum. The question is, in a cereal grain, the single cotyledon of the embryo is represented by, the correct answer is, the correct option is B, the scutellum. Then next question, the question number 15. Endosperm is formed during the double fertilization by, you know, endosperm is a nutritive tissue for the developing embryo. How it is formed? Option A, 2 polar nuclei and 1 male gamete. Fusion of 2 polar nuclei and 1 male gamete results in the formation of triploid endosperm. Option B, 1 polar nuclei and 1 male gamete. Option C, the fusion of ovum and the male gametes. Option D, the fusion of 2 polar nuclei and 2 male gametes. 2 polar nuclei, the option D, 2n plus 2n is equal to 4n. Ovum is diploid, male gametes are haploid, 3n. Then option B, 1 polar nuclei and 1 male gamete, n plus n is equal to 2n. The correct answer is fusion of 2 polar nuclei, fusion of 2 polar nuclei, n plus n, and 1 male gamete, n. So the correct answer for the question number 15 is option A, fusion of 2 polar nuclei and 1 male gamete results in the formation of triploid endosperm nucleus, the PEN, primary endosperm nucleus. Question number 16, Gemmule formation in sponges is helpful in, it's a kind of asexual reproduction seen in animals. Sponges comes under animals, you know. A, parthenogenesis, B, sexual reproduction, C, only dissemination, option D, asexual reproduction. The correct option for the question 16 is a kind of asexual reproduction, because in Gemmule formation of the organism sponges, fusion of gametes is totally absent. So asexual reproduction is the correct answer for the question number 16. Question number 17. Apomixes in plant means development of a plant. Already you read this. Apomixes means development of a new organism without the fusion of gametes. So the correct answer for the question number 17 is option B, apomixes, the development of a new organism or development of a new plant or development of a new animal without the fusion of gametes. Question number 18, pollination by wind. Wind means anemophilous kind of pollination seen in Nelumbium, Vallisneria, Eichhornia and Nymphea. Here pollination by wind. No, the question is incorrect. The pollination, question number 18, pollination by water is seen in, not wind, pollination by water. Pollination by water means hydrophily kind of pollination seen in Nelumbium, Vallisneria, Eichhornia and Nymphea. And Nelumbium, Nymphea are examples for attached floating hydrophytes. Eichhornia is a very good example for free floating hydrophytes. Vallisneria is a submerged hydrophyte. So water pollination or wind pollination seen in Vallisneria. So the correct answer for the question number 18 is option B, Vallisneria. Then question number 19, anemophilous pollination is mainly observed in, anemophily means wind pollination, the family option A, Gramineae, option B, Anonaceae, option C, Papilionaceae, option D, Euphorbiaceae. Anemophilous wind pollination generally seen in the Gramineae or the Poaceae. So the correct answer for the question number 19 is option A, Gramineae. Then question number 20. When a pollen tube enters the integuments, then the process is called, entry of pollen grain takes place through micropyle or through integuments or through the chalaza. The option is A, mesogamy, B, porogamy, C, chalazogamy, D, pseudogamy. If the entry of pollen grain takes place through chalaza, chalazogamy, entry of pollen grain takes place through the micropyle, porogamy. If the entry of pollen grain takes place through the integument, outer or the inner integument, it is said to be mesogamy. The correct answer for the question number 20 is option A, mesogamy. Then question number 21, the number of nuclei taking part in double fertilization. Double fertilization and triple fusion results in the formation of a diploid cell called the zygote and the primary endosperm nucleus. Option A, 2 nuclei, option B, 3, option C, 4, and option D, 5. Actually, at the time of double fertilization, one male gamete fuses with the egg, one nucleus, 2 nucleus. Then another male gamete fuses with the secondary nucleus. Secondary nucleus is the end product of fusion of 2 polar nuclei. So here 5 nuclei are involved. Number 1, the nucleus present in the egg. Number 2, the nucleus present in the male gamete. Number 3, the nucleus present in the polar nucleus. The nucleus present in the another polar nucleus. Number 5, the nucleus present in the second male gamete. Actually 5 nuclear fusion takes place at the time of double fertilization. And the correct answer for the question number 21 is option D, 5. And question number 22, endosperm is consumed by developing embryo in the seed of, endosperm is a nutritive tissue for the developing embryo. It is completely absorbed in option A, P, the Pisum sativum, option B, the maize, option C, the coconut Coccus nucifera, and option D, the castor resinus communis. The correct answer for the question number 22 is P plant, option A, the Pisum sativum, because at the time of embryonic development, the embryo completely consumes the endosperm, the nutritive tissue for the developing embryo. Question number 23. An example for naturally occurring parthenocarpy. Parthenocarpy means development of fruit without the act of fertilization. It results in the formation of seedless fruits. Option A, guava. Option B, mango. Option C, banana. Option D, apple. The correct answer for the question number 23 is, pretty well, option C, banana, the Musa paradisiaca. And dear students, these are all the important questions. Helps you to crack the NEET examination, national entrance, national NET, NEET, NEET, particularly NEET examination. So this national eligibility come entrance test. So NEET, these questions in the introduction of reproduction, flower, and pollination and fertilization. So read well, prepare well, you score very good marks in the NEET examination. Thanks a lot. Thank you.
இந்நிகழ்ச்சியை பற்றிய தங்களின் மேலான கருத்துக்களை கீழ்க்கண்ட முகவரிக்கு கடிதம் மூலமாகவோ மின்னஞ்சல் மூலமாகவோ குறுஞ்செய்தி மூலமாகவோ தெரியப்படுத்தலாம். அனுப்ப வேண்டிய முகவரி சிறப்பு அலுவலர், கல்வி தொலைக்காட்சி, 8ஆம் தளம், அண்ணா நூற்றாண்டு நூலகம், காந்தி மண்டப சாலை, கோட்டூர்புரம், சென்னை 6,85,000. தொலைபேசி எண் 7824015000.